Hi, everyone. My name is Floyd Nichols, and I am currently a third-year PhD candidate working with Maggie Osborne. So today, I'm going to talk a bit about my work in biosignatures in Mars analog hypersaline lakes. So many of you are probably familiar with the concept of hypersaline lakes, but for those who aren't, it's essentially any body of water that has a higher salinity than the ocean of 3.5%. And so with a lot of work in the hypersaline systems, we try to determine if life can really inhabit these environments. And most of hypersaline lakes on Earth are sodium chloride derived or sodium chloride dominated. And that's primarily just because of the ocean. And in these studies, we know that life is actually very viable in these salinities. However, we're not as familiar with some of the chemically diverse hypersaline waters, such as those that are calcium chloride derived, calcium sulfate, magnesium chloride, and even magnesium sulfate. Next slide, please. And so, actually, in a few studies that have been done in these chemically differing hypersaline systems, they sort of differ in terms of the life that is present or lack thereof in these environments. And so, in just a few studies that I'm showing here, we see that in magnesium chloride dominated environments, as well as magnesium sulfate acidic environments, life is not actually present or active in these systems. And so, that kind of poses a question of, does this occur in other chemical compositions, or even if we change pH, for instance? And so my lakes are magnesium sulfate hypersaline lakes, but instead of a pH of 1, they're slightly neutral to alkaline. Next slide. And so you can see here showing a ternary diagram of just the unique composition of the lakes that I'm looking at, shown here: Bass Lake, Last Chance Lake, and Salt Lake. We can see that in the cations, the dominant cation is mostly magnesium, and in our Last Chance Lake, shown by yellow, sodium. And again, sulfate is the dominant anion for our Bass Lake and Salt lakes, whereas bicarbonate is our dominant anion in Last Chance Lake. And so, we've got these differing compositions, and so we can sort of probe this question for what life might look like, or if there is life at all. And so, throughout the summer, I've just kind of been looking at lipids, which is a good way to look at biosignatures, as they can be diagnostic of life, although not as specific as DNA perhaps, but we can tell, at least generally, if it's microbial origin or plant origin or even human origin. And so, you don't really need to know the different categories here, but primarily we actually see a dominant microbial signature as shown by this sort of slate gray and blue bars here. And so, that was pretty exciting to see that in this environment, we actually do see a lot of life. And so next slide, please. And so I used a combination of isotopes as well as total organic carbon and total nitrogen to sort of provide further evidence that there is a microbial signature as shown here. And then, so some next steps is just kind of understanding preservation through geologic time. As shown here, I have total organic carbon with depth, and so hopefully I can pair it with lipids.